owls perform better on cognitive tests than early birds. Recently published research suggests that there is a link between a person's preference for morning or evening activity and brain performance. Scientists argue that people who declare themselves to be night owls usually have higher scores on cognitive tests. Scientists from Imperial College London analysed data from over 26,000 people to learn how various aspects of sleep, including duration, patterns, and quality, affect mental acuity and overall cognitive ability. Research has shown that people who prefer an evening-night lifestyle have better cognitive functions than people who start their activity at dawn. The results and description of the research were published in the journal BMJ Public Health. The data the scientists worked on came from the UK Biobank, an extensive database that has been maintained since 2006 and focuses on genetic predispositions and environmental factors, such as diet and lifestyle, to the development of the disease. Researchers analyzed information about adults who reported what time of day they felt most alert and productive and who completed a series of cognitive tests. They also checked their sleep patterns and other factors. Analyses showed that sleeping 7 to 9 hours a night was optimal for the brain, increasing cognitive functions such as memory, reasoning, and information processing speed. Sleeping less than 7 hours or more than 9 hours had a clearly detrimental effect on brain function. The researchers also found that a person's chronotype, or an individual's preference for evening or morning activity, also influenced test results. Night owls, i.e. people who are naturally more active in the evenings, performed better in cognitive tests compared to early birds, i.e. people who are active in the morning. The analysis was adjusted for other health and lifestyle factors such as age, gender, smoking, and alcohol consumption, and the presence of chronic diseases such as heart disease and diabetes. Younger people and people without chronic diseases generally scored higher. Healthier lifestyles were also associated with better cognitive performance. Our study found that adults who were naturally more active in the evening tended to perform better on cognitive tests than those who were active in the morning. These chronotypes may affect our cognitive functions, admitted Dr. Raha West from Imperial College London, the lead author of the study. This does not mean that all early birds have worse cognitive functions. The results reflect the general trend, she added. West also emphasized that it is important to remember not to sleep too long or too short. This is crucial for keeping the brain healthy and functioning properly. However, although sleep duration was important in the studies, people who reported problems with insomnia did not score significantly lower on cognitive performance. This may indicate that specific aspects of insomnia should be taken into account, such as its severity and duration. The duration of sleep has a direct impact on brain functioning. We believe that proactively managing sleep patterns is really important to strengthen and protect the way our brains work, said Daking Ma of Imperial College London, co-author of the paper. Not all specialists agree with the findings made in the publication. Without a detailed picture of what's happening in the brain, we don't know whether being a morning or evening person affects memory and thinking, or whether cognitive decline causes changes in sleep patterns, said Jackie Hanley of Alzheimer's Research UK. Jessica Cheloakis from Brunel University London added that the study had significant limitations because it did not take into account education or the time of day at which cognitive tests for the UK Biobank database were administered. Butter produced from carbon dioxide. A startup backed by Bill Gates has developed a method of producing fats from carbon dioxide taken from the air and hydrogen from water. There is no need to use animals, plants, or agricultural land in this process. According to the former head of Microsoft, butter produced using this innovative technology tastes as good as butter produced using traditional methods. Californian startup Savor, backed by Microsoft billionaire Bill Gates, claims to have developed a way to produce fats without the use of animals or plants. All you need is carbon dioxide from the atmosphere and hydrogen taken from water. Agriculture accounts for a significant share of global greenhouse gas emissions. These types of products can reduce emissions from this industry. The process releases no greenhouse gases, uses no agricultural land, and uses less than 1,000th of the water of traditional farming, Gates explained in a blog post. One of Savor's newest products is butter, which Bill says tastes like the real thing. It tastes really good, like the real thing, because chemically it is. I couldn't believe I wasn't eating real butter, the billionaire admitted. Synthetic production of dietary fats using chemical and biological processes has great potential. The raw materials for this method are the same as those used by plants: hydrogen taken from water and carbon dioxide from the air. The process is based on a concept presented last year in Nature Sustainability by scientists from the University of California, Irvine. Researchers have shown that fats from farm animals produce about 1 to 3 grams of carbon dioxide per 1,000 calories. But you can produce the same amount of fats in the laboratory with emissions of less than a gram. Large-scale synthesis of edible molecules using chemical and biological means without agricultural raw materials is a very real possibility. Such a method could avoid huge amounts of greenhouse gas emissions and at the same time protect biodiversity in areas that could be cleared for agriculture, said Stephen Davis of UC Irvine, lead author of the study, last year. Fats are made up of different chains of carbon and hydrogen atoms. These chemical building blocks can be obtained from water and air and then used biochemical processes to convert them into fats that are molecularly identical to those found in animals and plants. The new type of fat, the production of which does not require animals or large areas of land, is produced in a thermochemical system closer to the processing of fossil fuels than to food production. In this process, fat molecules can be built, creating chains of carbon dioxide, hydrogen, and oxygen. Researchers have developed a process that takes carbon dioxide from the air and hydrogen from water, heats them and oxidizes them, which separates the fatty acids and then creates fat. The result is real fat molecules like those we get from milk, cheese, beef, and vegetable oils, Gates writes on his blog. Kathleen Alexander, Savor's chief executive, admitted that the company's products could hit stores in a few years. We are currently in the pre-commercial stage and working to obtain regulatory approval to be able to sell our butter, she said. The startup also plans to develop similar methods of producing cheese, milk, and ice cream. The question is whether such butter and other synthetically produced products will gain recognition among customers. Getting people to give up their favorite dairy and meat products in favor of more experimental foods can be a challenge. Blocking one of the proteins extended the life of mice by over 20%. Blocking a protein that promotes inflammation extended the lives of mice by more than 20%. Moreover, inhibiting the action of interleukin-11 also reduced the incidence of cancer and other age-related health problems in rodents. This research gives hope for the development of effective anti-aging therapies. Scientists from the Medical Research Council Laboratory of Medical Science and Imperial College London, working with researchers from Duke News Medical School in Singapore, have found that blocking a molecule known as interleukin-11, IL-11, can extend the lifespan of mice, suggesting that agents that inhibit IL-11 may also have anti-aging effects in humans, because these proteins are also produced by our bodies. Drug candidates that block IL-11 are already being tested in humans in cancer therapy trials. The results and description of the research were published in the journal Nature. IL-11 belongs to cytokines, proteins involved in the immune response. These proteins have broad and complex functions. They can initiate inflammation, but also prevent it. They stimulate immune processes, stimulating immune system cells to react. Over 100 such proteins have been described so far, and new ones are constantly being discovered. Together they create a complex and extremely sensitive system. Scientists have been studying IL-11 for many years. In 2018, they showed that IL-11 is a pro-fibrotic and pro-inflammatory protein, overturning years of mischaracterization as anti-fibrotic and anti-inflammatory. Chronic inflammation contributes to aging-related diseases. As the body ages and accumulates damaged proteins and other molecules, the immune system often sees them as signs of possible infection. This can trigger inflammatory reactions that can cause further damage and contribute to cancer or autoimmune diseases. The link between IL-11 and aging was discovered by accident. Anissa Wijaya from Duke National University of Singapore Medical School tested a method for detecting IL-11. She accidentally included a protein sample taken from an old rat into her research. The test showed that IL-11 levels were significantly higher in this sample than in samples from younger rats. This project started in 2017 when one of our colleagues sent us some tissue samples for another project. Out of curiosity, I conducted some experiments to check IL-11 levels. It was clear from the readings that IL-11 levels increased with age, Wijaya said. The researcher admitted that increasing levels of IL-11 contribute to negative effects in the body, such as chronic inflammation, problems with wound healing or organ regeneration after injury. She also added that although the research was conducted on mice, the team hopes that their discoveries will translate into human health and life. This research is an important step towards better understanding the aging process. We tested a therapy on mice that could potentially prolong healthy aging and reduce the physiological symptoms of this process, she added. To test the effects of IL-11, scientists examined different samples from young and old mice. IL-11 levels were significantly higher in older individuals, in skeletal muscle, fat, and liver tissue. In the next step, they created mice lacking the interleukin-producing gene. This allowed rodents to live longer on average by more than 20%, compared to mice with normal IL-11 levels. The team found similar results when they used an IL-11 neutralizing antibody for 25 weeks in 75-week-old mice. That's roughly the equivalent of 55 years in humans. In mice administered the antibody from 75 weeks of age until death, life expectancy increased by 22.4% in males and by 25% in females. The mice lived an average of 155 weeks, compared to about 120 weeks in control mice that did not receive the antibody. In the video below, mice treated with an IL-11 neutralizing antibody are shown on the left and normally aging mice on the right. Both groups of rodents in the video are 90 to 95 weeks old. Moreover, rodents administered the antibodies had significantly fewer cases of cancer and diseases caused by fibrosis, chronic inflammation, and impaired metabolism, which are hallmarks of the aging process. Additionally, researchers observed few side effects. These discoveries are very exciting. The treated mice had fewer tumors and did not show typical signs of aging. We also observed a reduction in muscle atrophy and improvement in muscle strength. In other words, old mice receiving the IL-11 neutralizing antibody were healthier. Although the findings concern mice, it raises the tantalizing possibility that drugs that neutralize IL-11 could have a similar effect in humans. Anti-IL-11 therapies are currently being tested in human clinical trials for other conditions, which potentially offers exciting opportunities to study their impact on the aging body in the future, said Stuart Cook of Imperial College London, co-author of the paper. Life on Venus? Astronomers again detected phosphine on a neighboring planet and also found ammonia. In 2020, the world was informed about the discovery of traces of phosphine in the atmosphere of Venus. This gas is produced on Earth by some anaerobic bacteria, which has led to speculation about life on a neighboring planet. However, over time, more and more scientists began to question the validity of the sensational findings, pointing to data errors. Now scientists from 2 independent teams have found phosphine in the clouds of Venus again. They also found traces of ammonia. Scientists from 2 independent teams reported the discovery of phosphine and ammonia in the hot atmosphere of Venus. Both of these gases are referred to as biomarkers, which may suggest that some life forms exist in the clouds of Venus. The findings were announced a few days ago at the National Astronomy Meeting at the University of Hull in the UK. Venus is often called Sister Earth or Twin Planet. This is due to its size comparable to Earth, and the conditions there are far from those on Earth. The temperature on the surface of Venus is about 500 degrees Celsius. Venus is the closest planet to Earth and rotates on its axis in the opposite direction than most planets in the solar system. The planet has a very thick atmosphere and the pressure there is 92 times greater than on our planet. The idea of searching for microbial life on Venus is not new. Some models suggest that Venus had a climate suitable for liquid water to exist on its surface for up to 2 billion years. But it's more about the atmosphere of the twin planet, not its surface. Terrestrial microorganisms are able to enter the atmosphere. They have been found alive at altitudes of up to 41 kilometers by scientists using specially equipped balloons. This shows that life may be present in the atmosphere. As for the hellish conditions on Venus, there is a growing catalog of Earth microbes known to inhabit extremely harsh environments, including hot springs at Yellowstone, hydrothermal vents at the bottom of the oceans, toxic sludge in polluted areas, and acidic lakes. All over the world. The idea that microorganisms could live in the clouds of Venus was first proposed by astronomer Carl Sagan and biophysicist Harold Morowitz in 1967. A few decades later, David Grinspoon, Mark Bullock and their colleagues expanded on this idea. To check this, a series of probes was directed towards the twin planet, which showed that the temperature and pressure in the lower and middle part of the atmosphere of Venus, at an altitude of 40 to 60 kilometers, do not exclude the life of microorganisms. On the planet's surface, this would be impossible due to temperatures reaching nearly 500 degrees Celsius. But at altitudes of 40 to 60 kilometers above the surface, temperatures and pressures are closer to Earth's conditions and could potentially enable the survival of some very resistant extremophiles. The first reports about phosphine on Venus appeared in 2020 and immediately caused a sensation. However, since the announcement of the research results, this discovery has been questioned and experts have pointed out errors in the research. At a meeting of astronomers at the University of Hull, Researchers from Imperial College London reported the rediscovery of phosphine in the clouds of Venus. Phosphine, a gas also called phosphine, is a compound that is produced on Earth, outside laboratories, by anaerobic bacteria. This compound is also formed as a result of volcanic activity. In turn, astronomers from Cardiff University reported that they also found ammonia in the atmosphere of the neighboring planet. It is a gas that was first detected on Venus in the 1970s. On Earth, it is produced mainly by biological activity, but also by industrial processes. Its presence on Venus is difficult to explain. Scientists know of no chemical process on Venus that could produce this compound. The presence of these two gases in the Venusian atmosphere is not conclusive evidence that some form of microbial life exists there. However, it ignites the imagination. If Venus had gone through a warm, wet phase in the past, as global warming occurred, life would have evolved to survive in the only niche it had left, the clouds, said Dr. Dave Clements of Imperial College London at the meeting. New observations by Clements' team using the James Clerk Maxwell Telescope, JCMT, in Hawaii have revealed the existence of phosphine in the atmosphere of Venus. By tracking its signature, scientists saw that phosphine appeared to follow the day and night cycle on its twin planet. Our findings suggest that when the atmosphere is bathed in sunlight, phosphine is destroyed. All we can say is that phosphine is there. We don't know what produces it. Maybe it's the chemistry we don't understand. Or possibly life, Clements noted. There are many oddities in the atmosphere of Venus. Phosphine is just another one of them. Among other strange things is the way the amount of water and sulfur dioxide in the atmosphere changes over time. It's unclear why, Clements added. It's similar with ammonia. Astronomers emphasize that it is difficult to explain its presence in the clouds of Venus using known atmospheric or geological phenomena. Its traces were discovered by researchers from Cardiff University using the Green Bank telescope. They suggested that if life exists on Venus, it could use ammonia to neutralize the sulfuric acid that makes up Venusian clouds, making tiny water droplets habitable. If there are any microbes in the clouds of Venus, they may produce some gases that we would not expect. That's where ammonia comes in because it can be used as a way to neutralize acid, said Professor Jane Greaves from the University of Cardiff. Greaves led a team of researchers that caused a sensation in 2020 with references to phosphine on Venus. These are very exciting findings, but it is important to emphasize that these are preliminary results and further work is needed to learn more about the presence of these two potential biomarkers in the clouds of Venus. Nevertheless, it is fascinating to think that these discoveries could point to possible signs of life or some unknown chemical processes. It will be interesting to see what the research reveals in the coming months and years, said Robert Massey of the Royal Astronomical Society.